So, welcome to Genomics of Cancer. This is a very big topic. This is a place where genomics can absolutely be applied in nursing practice. And it will sort of take that information that you learned in your pedigree assignment and make it a little bit more real. So, what are our learning outcomes? In this unit, we're going to understand the genetic basis for cancer. That includes both acquired mutations in the somatic cells and hereditary cancers that are in the germline and get passed from one generation to another. We'll examine the role of mutations in oncogenes. Those are the accelerator genes that tell the cell to make more copies, suppressor genes that silence that process, and DNA repair genes that identify defective cells and target them for cell death. Mutations in those genes are implicated in development of many cancers. We will differentiate characteristics of normal cells from benign tumor cells and malignant cancer cells. And then we'll compare the processes of initiation and promotion. And then we'll look at progression and metastasis in cancer cells. Lastly, we're going to discuss the role of epigenetics and aging in relation to cancer and why that's important. And we will analyze some family pedigrees and discuss the concept of a sporadic cancer versus a familial cancer. Sporadic cancers account for at least 80% of cancers. Familial cancers are those that are inherited, 5 to 15%. I know that leaves some unaccounted for. And it's just because we don't know that information yet. So why is it important to study genomics in cancer? Well, cancer is very prevalent. In the United States, your risk over a lifetime of acquiring cancer is about 39.1%. It's going to cause a significant amount of morbidity and mortality. In the year 2020, which is the most recent statistic I could find at the time of this presentation, cancer was responsible for more than 600,000 deaths in the United States. Understanding how genetics is involved in cancer helps healthcare providers identify individuals at risk for developing cancers, and it helps us detect and diagnose cancer with precision. The earlier the better. Cancer is a race against time. As those cells begin to rapidly multiply and become more abnormal and more invasive, we lose our advantage in treating it. So identifying risk and detecting early is our best bet for a healthy outcome for that person. And finally, genetics or genomics can be used to treat cancer. You can base treatment both on the client's inherited genotype. I may attach a video to the page for this unit, optional to look at it, but talks about treating people with chemotherapeutic agents based on their response to medications. And we can also use the cancer's genotype, which is going to be significantly different from the original genotype that the person inherited. Some cancers are responsive to estrogen, so HER2 blockers might be effective. Other cancers are not, so those agents would not be effective. Knowing what that cancer cell's genotype looks like a lot of times can guide treatment decisions. So this is important to understand. So there's a little graphic of cancer. You see the nice healthy cells right here. And here are the cancer cells. They look very different. We're going to talk about characteristics of cancer cells. All cancer is genetic. Interesting statement. But only 5 to 15% of cancers are considered hereditary. So what does that mean for us? It means that all cancer is genetic and that it's always caused by changes in the expression of genes, those oncogenes, suppressor genes, and DNA repair genes. Please remember those. We'll talk about them in more detail throughout this presentation, but those are the three types of genes that are most often implicated in cancers. All cancer is caused by changes in the expression of those genes. Cancer cells transmit those changes, those mutations, from one cell generation to another. But cancer, strictly speaking, is not inherited from one human generation to another. Even in familial cancer syndromes, the person isn't born with cancer. You very rarely see newborns with cancer. Once in a while it happens, but it happens during fetal life as in certain very rare aggressive types of cancer. But it is always like an acquired change. Nobody is, the genotype doesn't give you the cancer. It's the changes. Um, Cancer happens when normal cells are altered at the DNA level. Um, we'll sort of parcel that out as we move along. Hereditary cancers, those cancers that run in families, where we say you have Lynch syndrome or Fanconi syndrome, um, these are a result of weaknesses in inherited DNA that greatly increase the risk for abnormal cell division. So, oncogenes, suppressor genes, and DNA repair genes may have weaknesses. So, the body is trying to keep order in the cells, and it does a really good job until it gets overwhelmed. And if there are hereditary weaknesses in the processes that regulate healthy cell division, that's when we see cancer. So, some evidence, this is just background information. This supports the link between genetics and cancer. How do we know that cancer has a genetic background or basis? So, people with chromosomal anomalies like Down syndrome or Turner syndrome have higher incidence of certain cancers because genes on those chromosomes regulate healthy cell division. People with broken or fragile chromosomes like Fanconi syndrome, people with different copy number variants, also have higher incidence of certain cancers. Some cancers can be very prominent in a person's pedigree, and it can be shown that they're inherited in an autosomal dominant pattern, and that looks a certain way. If you go back to week 5's content, was it week 5 or week 6? When I talked about the pedigree, I showed you some examples of pedigrees that revealed an autosomal dominant pattern for diseases. So some cancers appear to be inherited in that way. Families do seem to have, some families have a higher incidence of a particular cancer, like a colorectal cancer, breast cancer, ovarian cancer, than the risk that's seen in the general population. For example, with breast cancer, not all breast cancer is familial or considered to be familial. Your lifetime risk, if you are a woman, is about 1 in 8 of developing breast cancer from any cause. If you have a familial hereditary breast and ovarian cancer syndrome, like the BRCA1 gene mutation or the BRCA2 gene mutation, you will see that people in your pedigree have a higher incidence than 1 in 8. It might be, out of the women in your family, 1 in 4 or 1 in 5 have breast or ovarian cancer. We know from looking at cancer cells under a microscope that cancer cells generally have an abnormal number or structure or both of chromosomes. So aneuploidy in cancer cells, extra chromosomes, missing chromosomes, very, very common. So things just don't go right and nothing edits that process. The more advanced or aggressive a cancer is, the less similar it is to the parent cell in terms of its DNA. So the person's genotype, when you sample normal cells, will look like maybe 46XX. If you look at a cancer cell, it might be 50XX, or it might have more. And the more advanced a cancer is, the less its DNA looks like its parent, that original cell. So, processes that are involved in the development of cancer include basic DNA structure and function, control of cell division, reproduction, and apoptosis. Cancer cells have sort of an immortality. They do not die. That is what makes them so dangerous. They divide at a higher rate than they should, which is what grows tumors, these masses of cells that aren't needed for function, and they reproduce more frequently. Errors in protein synthesis can lead to certain types of cancers. Cancer cells will also start to synthesize different types of proteins. We'll talk about all of this in more detail. Don't think you have to really memorize this slide. Just understand how these different cell functions are involved. Mutational events. So when you are exposed to a carcinogen, let's say it's UV sunlight, it can change genes, especially if those chromosomes are a little bit fragile to begin with. And that leads to abnormal production or overexpression of oncogenes underexpression of suppressor genes. Lastly, we have Mendelian inheritance, and that's implicated in family history when we look at a pedigree and see the patterns of inheritance, like autosomal dominant. Some cancers are associated with syndromes that are autosomal recessive, but most cancers are due to those mutational events. Acquired mutations and cancer. So these are responsible for most of the cancers that are diagnosed. These mutations occur in somatic cells. So they can happen in a skin cell, they can happen in the cells that make up breast tissue, but something happens locally at that small level, and it causes abnormal cell characteristics. And this happens because of damage to cells from environmental exposure. These environmental agents are called carcinogens. So tobacco is a very notorious carcinogen, tobacco smoke and all of its byproducts. There are many chemicals in cigarette smoke that induce changes at the cellular level. They damage that DNA. UV light, so sunlight, we know, causes changes in the DNA. Ionizing radiation. So, for example, Hiroshima, when there was a nuclear explosion, people were exposed to massive amounts of radiation, and they quickly developed different types of cancers, especially leukemias. And you saw it throughout the lifespan. So that carcinogenic effect of that radiation caused rapid, massive damage to cells that overwhelm the body's defenses. Viruses like HPV is implicated in cervical cancer, certain types of oral cancers. Exposure to that virus damages the cell beyond what the body can manage and can cause cancer. Aging. So the aging process involves cellular senescence. The DNA becomes a little less stable as the telomeres get shorter. We are going to discuss aging in a separate presentation. It's important to understand that. But aging itself is sort of a carcinogenic event. So these changes aren't found in every cell of the body. They're found in some cells and not others. They are not passed down to offspring. And that is responsible for most of your cancer. Inherited mutations occur during gametogenesis. They're in the germline. They could have been there for many generations. They increase risk for cancer substantially above what the general population experiences. These mutations will occur in every cell, and they can be passed down. They will usually lay dormant until something happens to damage that cell. So these are the types of genes that are affected. The first three are the most important. Then we have immune genes at the end. Oncogenes, they function like the accelerator on a car, and they say go, 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 go. They tell the cell it's time to go into that cell cycle, into mitosis, and reproduce. They are used to replace non-functional cells and to grow when that is necessary. So children have a lot of oncogenic activity, but it's healthy and normal because they need to grow their organs or tissues. That's time of rapid growth. But all of us have to replace our skin cells, the cells that replace our intestinal linings. Most cells in the body do have some sort of replacement. Some do not. And we'll talk about that in a second. Overexpression of oncogenes can lead to cancer. For example, HER2 /NEU is a type of breast cancer that is caused by overexpression of oncogenes. The genes are dividing too rapidly and mistakes are made. And the mechanisms in the body that will target those genes are overwhelmed by all of this rapid growth. Abnormal oncogene activity is more likely to be environmentally induced. If you go back to the unit where we discussed epigenomics and you talk about the ghost in our genes, that movie that I kind of required And I hope you had time to watch it. It is certainly worthwhile. Um, most of the time, these oncogenes are silenced with little methyl tags, and the methyl tags keep the oncogenes from overexpressing themselves. Environmental exposures can demethylate oncogenes so that all of a sudden they're expressing themselves when they would normally be silent. So, environmental interaction is important in that process. Suppressor genes act like brakes on a car, they're the opposite. So, there's always this dance in the body. How much growth versus, let's keep this under control. They act like the brakes and they inhibit cell reproduction. They also send chemical messengers to cells when they get old and they're no longer functional and tell them to die. So the cell commits suicide. It's kind of a grim metaphor, but it basically is what happens. And this is a healthy process. It keeps defective cells from reproducing. If their DNA is likely to be damaged and mutated, um, the healthy suppressor gene activity should tell them to die. It's time to go. Under expression of suppressor genes, people with abnormal um, defects in suppressor genes that are underactive can lead to unchecked cell proliferation and cells that do not want to die. Those are more likely to be cancerous. Defects in suppressor genes can be hereditary, or it can occur as methylation events or changes in the histone modification system. There can be an environmental factor there, but we need suppressor genes to prevent unlimited growth of cells that aren't necessary for function. DNA repair genes fix mistakes. So they kind of scan the cell. Do you look like what we want? Are you the right candidate for this job? And if they have any genetic abnormalities, a DNA repair gene should fix that mistake. Either it can directly act on the genome to cut out the mistake, or it can just program the cell to die. BRCA1 and BRCA2 are repair genes. So mutations on BRCA1 and BRCA2 are responsible for breast and ovarian cancers associated with that syndrome, that familial cancer syndrome. There's a defect in recognizing an abnormal cell and targeting it for repair or elimination. Hereditary colon cancers also. So Lynch syndrome is another syndrome we're going to talk about at the end of this presentation. They are caused by defects in DNA repair genes as well. Immune genes. Immune factors can recognize and destroy cancer cells. Any defect on the HLA genes, for example, those genes that are responsible for killing invaders, impair the ability of the body to eliminate mutated cells. Okay, we're going to take a break here and we'll pick up on the next slide